Next, we have An Hodac from chemistry, and he will be presenting on biocatalyst efficiency enhancement through bioengineering.
Hi, so my name is An Hodac, and I work as a research scientist in Dr. Hati's biochemistry research lab. So, do you know how long it takes bacteria to become resistant to antibiotics? Well, a study done in Harvard shows it only takes 11 days for a strain of bacteria to become resistant to antibiotics that would have previously killed them. Imagine a world where antibiotics or drugs that are developed have no impact on diseases. That's a very dangerous and realistic world we live in. Just keep that in mind. But my research focuses on understanding how this enzyme called prolyl tRNA synthetase, also known as ProRS, but don't worry about the name. Just think of this as an enzyme that plays an important role in making the building blocks of life within all living organisms, from humans to bacteria. So, the focus of my talk will be presenting on the applications of my research in fine-tuning the capabilities of ProRS and to further understand how it works within the bacteria E. coli, because it's a good model organism and it's very accessible. So, in my lab, I alter the DNA of this enzyme, so when it's expressed as when it's expressed as the enzyme, its function changes compared to the original form. So then from there we can understand how it functions. So DNA is interesting in the fact that there are bits and parts of it that are more or less relevant or important to the end product of the enzyme. So if we mutate or change the important DNA, then the resulting translated enzyme's function also changes. From there, we're able to understand which parts of the enzyme is important and relevant to its function and structure. So here is a model of the enzyme I work with, very, very large model. And the chemistry usually takes place right at the center. But the focus of my research is on these peripheral clefts right here to see if they are important or relevant to the function of the enzyme. And our research has shown that once these clefts are removed, it actually destroys the overall function of our enzyme completely, so they are relevant. Now, what's the point of this whole talk, and why was I talking about drug-resistant bacteria earlier? Well, that's because what I didn't mention is that even though this enzyme is inherently present in all living things, its structure is inherently different between humans and bacteria. So again, this model is of the bacterial form, and the human form of this ProRS model doesn't have these peripheral clefts. So with more research, we are able to design drugs that target these peripheral clefts in the bacteria and able to design drugs that the bacteria cannot become resistant to, since this enzyme is crucial in the survivability of bacteria, and therefore solving a worldwide health problem. Thank you.